Insulin and glucagon are key hormones in maintaining control and homeostatic levels of our blood's glucose concentration. Our pancreas secretes both insulin and glucagon based on whether or not our blood glucose levels are too high or too low. Insulin and glucagon are key hormones in maintaining control and homeostatic levels of our blood's glucose concentration. Our pancreas secretes both insulin and glucagon based on whether or not our blood glucose levels are too high or too low. In order to transport glucose throughout our body, specifically within our bloodstream, the red blood cells take up facilitated diffusion. Facilitated diffusion allows for glucose within our body to travel passively from an area of high concentration to low concentration, with no energy required. Our pancreas is both an endocrine and exocrine organ. Within the pancreas, the endocrine version contains clusters within the cells known as pancreatic islets, islets of Langerhans. Beta cells of the pancreas secrete insulin in order to decrease high blood sugar, whereas alpha cells of the pancreas secrete glucagon in order to increase blood glucose levels. In other words, when we have high blood sugar levels, beta cells secrete insulin. And when we have low blood glucose levels, alpha cells secrete glucagon in order to return our blood sugar/glucose levels back to homeostasis. In order to understand how these hormones maintain a homeostatic blood glucose level, negative feedback must be a comprehended process. Negative feedback can best be described as a process in which an imbalance within the body is corrected in order to bring levels of imbalance back to a normal homeostatic level. For example, when blood glucose levels are disturbed, the pancreas acts as a negative feedback mechanism, returning balance. Both insulin and glucagon have significant effects on their target cells throughout our body. Insulin, in fact, tends to speed up and increase glucose uptake within all of its target cells. GLUT4 transporter is the insulin-regulated transporter that is responsible for the disposal of glucose. This specific transporter can be found in the tissue of the heart, skeletal muscle, and adipose tissue throughout our body. Insulin is also important because of the accelerated ATP production, as well as glucose entering the target cells, specifically within the liver and the skeletal muscle. When there is an overflow of glucose within the liver and skeletal muscle, these regions of the body store this as glycogen. Insulin is also key in stimulating amino acid absorption and protein synthesis. The last important characteristic to insulin and its effects on its target cells would be the acceleration and stimulation of triglyceride formation within adipose tissue, or our fat. All the effects of insulin on its target cells throughout our body are key to maintaining our health and allowing for our glucose concentration within our blood to be stable. Although glucagon does not seem to have nearly as many effects, these results are just as important as the role of insulin. When released, the glucose molecules are either rapidly metabolized for energy within our skeletal muscle fibers or released within our bloodstream thanks to the cells of our liver. As said before, insulin stimulates the formation of triglycerides within our adipose tissue. Glucagon, however, stimulates the breakdown of these chemicals in order to be sent through our bloodstream to be used by other tissues within our body. Glycogenolysis is a process that takes glycogen and breaks it down into glucose. In order for glucose to be synthesized and absorbed, glycogenolysis must occur. Glucagon is also used to produce and release glucose from the liver to be absorbed with amino acids and recirculate back to the bloodstream. This synthesis of glucose in the liver can also be referred to as gluconeogenesis. Along with gluconeogenesis, the process of glycolysis is crucial when discussing the control of blood glucose concentration. Glycolysis can best be defined as the breakdown of glucose into molecules of pyruvate, with the finished product and gain of 2 ATP molecules. Where and when glycolysis occurs helps determine rising or dropping insulin and glucagon levels. In some cases, all of these processes and steps to prevent a homeostatic imbalance can fail and can escalate to a variety of diseases. Within the body, if there is an excessive amount of glucose within the bloodstream, a patient will have hyperglycemia, which refers to chronic high blood glucose levels. Conversely, if there is a significant deficiency of glucose within the bloodstream, someone might have a disease known as hypoglycemia. Both conditions can be derived from diabetes or can be a sign of future diabetes or diabetic complications.